Good morning, good afternoon, and good evening, everyone, from wherever you're joining us, and welcome to the 10th webinar in the Women's Health 2024-2025 series. Today's webinar is titled Challenges and Opportunities for Treatment of Metastatic Breast Cancer, featuring Dr. Marsha Rosner, professor at the University of Chicago. Today, Marsha will present some of the key challenges faced in trying to eliminate metastatic breast cancer and some novel approaches to addressing these challenges. Before we get started, I would just like to acknowledge our partners for this series, the American Physiological Society, the Society for Women's Health Research, and Scientist.com. We'd also like to say thank you and give a huge shout out to all of our series sponsors. None of this would be possible without them. I'm Sarah McFarland from the events team here at Scientist.com, and I'm happy to be your host for today's event. Before I welcome our speaker to the floor, I just wanted to cover a few housekeeping notes to help you get the most out of your experience today. First, take a look at the interactivity panel at the right of your screen with 4 tabs: chat, Q&A, polls, and handouts. This is where you'll participate in the Q&A as well as access all webinar resources. We want this to be an interactive experience for you, so please send your questions, thoughts, and comments using that Q&A tab, and we'll try to address as many of your questions as we can at the end of the webinar today. You can also explore some links in the handouts tab to learn a little bit more. And lastly, if you're having any technical issues, please try refreshing your browser and ensure that you're using Google Chrome to access this event, as it is the most stable browser. And if you're still having troubles after you refresh, use that private chat function to communicate with me by sending a chat to Sarah, and I can help you to troubleshoot. All right, and now I'm very pleased to welcome Dr. Marsha Rosner to the floor. Marsha, take it away whenever you're ready.
Okay, so thank you so much, Sarah. It's a pleasure to be here, and I'd really like to thank the organizers for the invitation. So cancer is the second leading cause of death in the United States after heart disease. And for example, in 2021, there are 1.9 million new cancer cases and over 600,000 cancer deaths. So as you can see, there's actually been a pretty dramatic drop in male incidence, slightly less in female incidence. But this recent drop in cancer deaths is largely due to decreased smoking and improved early detection and treatment. So despite this good news, of the top 4 cancers, the long-term decline in mortality has halted for prostate and slowed for breast and colorectal cancer. So there's more for us to do. For breast cancer in particular, which is the subject of my talk today, it is the leading cancer in women. About a third of women will actually contract breast cancer during their lifetime. And it's the second cause of cancer death in women. Again, about 15% of women will succumb to breast cancer. So as you can see, there are many other cancers, and each of them is problematic. Perhaps the most in terms of death, as noted before, is still lung and bronchus, and again, largely due to smoking. What I'm going to talk about today is a particular subtype of breast cancer called triple-negative breast cancer. It's the most aggressive breast cancer subtype. And so, as you can see, there are several subtypes of cancer, going from luminal A, normal-like, luminal B, HER2 enriched, and finally triple negative. And in that order, they become more and more aggressive to the point where the survival changes significantly. So how do we distinguish between these cancers? Largely, it's based on receptors. First, hormone receptors, the estrogen receptor, and the progesterone receptor, which are steroid receptors, and HER2, which is the tyrosine kinase receptor. So luminal A, normal-like, luminal B, they have hormone receptors, estrogen receptor, progesterone receptor. HER2 enriched may or may not have the hormone receptors, but they're highly enriched in this tyrosine kinase, which is a driver for that particular type of breast cancer. Triple negative is distinguished because it's negative essentially for all 3. Doesn't have estrogen receptor, doesn't have progesterone receptor, and it's low in HER2. As you can see on the right here, Triple negative is particularly bad for certain classes of people, particularly African Americans, and also those who come from economic hardship. And so compared to European Americans, African Americans are much more subject to triple negative breast cancer. But what's particularly alarming, as you can see, is this is a disease that's becoming more and more prominent among younger people. And actually, this is true of a number of cancers, such as colon, and we don't actually know the reason for that. But it makes it more important to address what's going on here. So the good news is the breast cancer 5-year survival rate is about 91%. But now if we look at triple negative breast cancer, the 5-year survival rate is overall 77%. It's not all bad, because if your disease is localized around the primary tumor that starts, then the survival rate is 91%. But as the tumor or the cancer itself becomes more distant from the original cancer, regional is 66%, and distal, which I'll get to in a minute, is only 12%. So this is a disease that we really need to focus on. So the question I want to address here is, why is cancer so challenging? And these are my answers. It's a complex disease. And the first reason is it's not one disease. It's actually multiple diseases. As we just saw, there are differences in tissue and differences in subtypes, but there are even differences between individuals. So if you have breast cancer and I have breast cancer, our cancers are going to be different. But what's more important and potentially even more alarming is that even within one tumor, let's say I have breast cancer, I have a tumor, the cells are different within the tumor. Most cancers have complicated origins. So we hear about genetic mutations, but this is not true of many cancers. We don't even know how they started. What is also particularly unique to cancer is it's an evolutionary disease. It changes over time. It adapts to changes in the local microenvironment. And again, I'll get back to this a bit later. And finally, if one's talking about solid tumors like breast cancer, then the dissemination of tumor cells throughout the body by metastasis is the primary cause of death. So the tumor may start in a primary place, but then the cells can actually begin to move, migrate, invade through the connective tissue, intravasate into blood and lymph vessels, where they migrate, get transported, they extravasate outside to different tissues, where they can colonize, proliferate, and actually introduce blood vessels to give them a source of nutrients at a secondary site. This is the major challenge in solid tumors, metastasis. It's characterized by many distinct biological states that are driven by stress in the environment. Don't have nutrients, don't have oxygen, so the cells, as they keep growing, are stressed, and they cannot stay in that environment, and they need to move out if they're going to survive. Metastatic cells are drug resistant, but the mechanisms by which this occurs are poorly understood. But the key feature is that, as I alluded to before, they're heterogeneous. This can be on a genetic level, an epigenetic level, and by epigenetics, what I mean is the way genes are transcribed from the DNA to the RNA. And there are a lot of modifications that can occur in histones and other modifiers of the transcription machinery that will change the expression of those genes. And then phenotypic. And I'll get to later, phenotypic could be just whatever one sees as a function of a tumor. Maybe that comes genetically, maybe that comes epigenetically. Maybe that's just stochastic. It just comes from some change in the environment that the cell is responding to. So we would suggest that understanding the regulators of metastasis, how metastasis is actually regulated, is key to generating successful therapeutic interventions. So what are the drivers of breast cancer metastasis? Well, as I said, genotypic drivers related to genetic drivers of cancer, largely resulting from mutations in key genes, are the ones that we've really focused on the most. And for breast cancer, there's specific drivers which lead to common pathways to target. And I've mentioned two, these are the major drivers, the hormone receptors, particularly the estrogen receptor, ER+, and the kinase, tyrosine kinase receptor, HER2, which is part of the EGF receptor family. And so these are targetable, but they also can mutate and become constitutively active. In addition, along the signaling pathways that emanate from the action of these receptors are other players, such as AKT, another kinase, and these can become mutated. And so they often are targeted by our drugs. Again, these are common signaling pathways. So how well are we doing in terms of these targeted cancers, these targeted genes in the cancers? And the answer is we've had mixed success. So we wouldn't say that we're curing cancer, but we now feel we can do long-term management, particularly of these hormone receptor cancers, the estrogen receptor positive, and some of the HER2 positive tumors because of this targeted type of therapy. But then there's another driver of breast cancer, and here I'll call it phenotypic. And this, what I mean by that, it's non-genetic. We're not being driven by mutations at the level of DNA. And these can be transient, such as I said, some kind of change in the environment, or I'm going to fold under here the epigenetic changes that can occur. Epigenetic can come later on from genetic changes, but also phenotypic. And when they occur, they can actually be heritable. So they can go from generation to generation of cells. And these often occur in response to stress. So our cells have very distinct stress responses, and they respond to lack of oxygen, nutrients. They can even respond to drugs and changes in the immune system. So what happens when that occurs? That causes changes in tumor cell states. So originally, most of the solid tumors we're talking about have a so-called epithelial type of cell type, which tends to be skin-like, lots of barriers. They don't really move. And during cancer, when they're under stress, they can actually change and move into a more mesenchymal type of cell, which is characteristic, let's say, of fibroblasts, which move in when we have wounds. So they're the motile type of cell. And this is very important for cells that eventually have to move, invade, and become metastatic. So phenotypic changes can cause changes in these tumor cell states and drive them from an epithelial to metastatic state. And in addition, they often end up not exactly mesenchymal, not exactly epithelial, but they have characteristics of both, as well as lineages that may not even relate to breast cell lineages. They actually may come from other kinds of lineages, like neuronal lineages. So cancer cells can actually change their nature, their whole character. But what's the key problem in all this is that there is no common pathway to target. So we don't have estrogen receptor specific drugs. We don't have HER2 specific antibodies. And as it turns out, this type of driver of breast cancer is the primary driver of triple negative breast cancer, but also the primary driver of metastasis, which is why triple negative breast cancer is potentially so lethal. And as I alluded to before, the survival overall is relatively poor. So how does this play out in clinical trials? Well, even though clinical trials for breast cancer in general are pretty positive, as I suggested, with the 5-year survival rates, for metastatic breast cancers, they're basically stagnant, and the patients have not really shown a lot of significant improvement. So I think I'm lucky to have a clinical fellow in my laboratory, Marguerite Matosian, who's on the front lines of cancer care. And she's the one really who walked me through the types of treatments that we have. So There's good news and bad news of all of this. But basically, we have 3 lines of defense. So initially, and traditionally for triple-negative breast cancer particularly, we use chemotherapy. One of the new kids on the block is so-called immune checkpoint therapy. And that means that we can block essentially the exhaustion of T cells that are trying to attack tumor cells by using blockade therapy. And this enables these T cells to be reactivated. And this was very exciting for triple-negative breast cancer for a while, but basically the benefits of this type of treatment at this point have plateaued. So we're looking for other ways to address the problem. And then if you have a particular mutation, if one has a particular germline mutation called BRCA1 or 2, which relates to DNA repair pathways in cells, then one can actually target the protein PARP, and this has also been an effective treatment mechanism. So that's line 1. If that doesn't work, then I would say the newest approach, the one that's causing the most excitement, are antibody drug conjugates that deliver different kinds of drugs, and in particular, delivering chemotherapy, such as doxorubicin. So basically, antibodies can grab onto proteins sitting on the surface of cancer cells, and they're attached to a chemotherapeutic drug, which then, upon hitting the cell, can be delivered more specifically into the cell. So it's kind of a targeting, a localizing of that therapy to tumor cells as opposed to other cells in the body, which can minimize overall toxicity and secondary effects and essentially increase the efficacy of any drug treatment that you have. But again, this is limited to, although it has a lot of potential, a lot of trials are underway right now, so we don't even know how long this therapy will last. And this leads to presence as a third line. If we have a biomarker, then we can go back to targeted therapy, and if we don't, then we can use chemotherapy. So how is this used? Well, as I mentioned, there's lack of significant progress from the treatment, particularly for TNBC. And the reason is because we don't really have inhibitors of biomarkers that are specific for TNBC. So on the right are a set of drugs. And as you can see on the top, if you're targeting HR positive or HER2, there are a lot of specific targeted treatments that we can try. But the other treatments that aren't specifically targeted can be for any tumor, but we don't have one just for TNBC. And so what usually causes people to succumb is infection and organ failure as the most common causes of death. And so at the end of the line, we don't really have a cure for metastatic breast cancer, but to a large extent, we're beginning to learn how to control it. So why? Why is that true? And we would suggest that the reason is this heterogeneity, and not just a kind of static heterogeneity, but a heterogeneity that's dynamic. As I said, this is an evolutionary disease, so this disease keeps changing over time. Frankly, killing cancer cells is easy. I could do that tomorrow. If you came to me with a patient and said, I can kill all of your cancer cells, but I probably kill you too because I'm killing normal cells at the same time. So what can we do? We can try to selectively kill malignant cancer cells, as I've just suggested from the previous slide, or we would suggest maybe we should think about reprogramming cancer cells, essentially resetting them so that we eliminate the metastatic disease. We eliminate the signals in them that are driving them to metastasis. We reduce the heterogeneity, and then we can more effectively kill the non-metastatic cells. So how can we do this? So today I'd like to address 2 key questions. The first is, what do we know about how metastasis is regulated? Can we actually identify some of these regulators? And then what might be some optimal strategies for inhibiting metastasis? How can we think about it differently? So in our lab, what we focused on are actual natural physiological regulators of metastasis called metastasis suppressors. So many people have known about tumor suppressors such as P53, whose natural role is to block tumor growth, very similar to, let's say, a brake in a car. If the accelerator is on and you want to shut off the car, you push on the brake. Metastasis suppressors are proteins that do similar things, but they target metastasis. Sometimes they'll affect tumor growth, but not always. Often the tumor can be very happily growing, but these suppressors block the ability of tumors to metastasize to a different site. So they're physiological proteins expressed in healthy cells and local disease, but like tumor suppressors, they're lost in metastatic disease. And where do they act? So from this slide, you can see from the primary tumor, all of these steps: invasion, intravasation, migration and transport, extravasation, and colonization. They can block, different suppressors can block different aspects of these steps. And the one I'm about to talk about, which is called RCAP, blocks essentially the movement from the primary tumor to the point of colonization and growth at a secondary tumor site. So this suppressor, RAF kinase inhibitory protein, or RCAP, is a very, very conserved protein. As you can see on the right, this is its family tree. What it's technically called, the gene name, is phosphatidylethylamine binding protein, or PEBP. Again, a misnomer like many of the names that we use in biology. And this is actually conserved not just through evolution, but as a sequence, but structurally. As you see on the left, it's a very compact spherical protein. And in mammalian cells, it's also regulated by phosphorylation of the serine 153. So you can actually make it a more potent actor, in the good sense of being a suppressor, if you block its ability to be phosphorylated at that site. So what does it do? It's a homeostatic regulator of the kinome. What does that mean? If the kinome's perturbed, kinome meaning kinases in the cell, these are signaling agents in the cell, and let's say they're overactivated, this can help to shut them down. Turns out it's actually a regulator not only of cancer, but of heart disease. It's a major player in both. It's the phosphorylated form at this S153 is the part that actually works in heart disease. And in both cases, it's a good guy. It helps to suppress heart disease. It helps to suppress cancer. And as I noted specifically, its role in cancer is as a metastasis suppressor. So on the right, this is an experiment we did with breast cancer, where we injected triple-negative breast cancer cells. They metastasized to what looks like the brain, but it's actually the skull. So it's a bone metastasis. And if we introduce wild-type RCAP into our cells, we can suppress that hugely. And if we actually make a mutation at that serine site so that we can't phosphorylate it, in fact, it's almost complete. And at the same time it's changing the cancer cell, it's also changing the ability of that cancer cell to reprogram the tumor microenvironment, and particularly macrophages, called tumor-associated macrophages, that enter into the tumor and actually facilitate the ability of those cells to move and to enter into blood vessels and metastasize. So it's a good guy on multiple levels. But the way that we've used it is also to say, who is it regulating? How is it working? And one of the players that we've identified is a protein called BOC1. And BOC1 is the transcription factor. So as you can see, RCAP, as I suggested, is an inhibitor of kinases. But these kinases actually activate BOC1, and BOC1 then can lead to transcription of genes that do multiple things, we now know, but one of them are genes that promote invasion and lead to metastasis. So BOC1, which other term is BTB-CNC, homology 1, basic leucine zipper transcription factor 1, forms a heterodimer with other proteins called MAFs. And normally it's a repressor when it's associated with MAF, and it competes, those who know it, another transcription factor, NRF2, NRF2, which is an antioxidant. It turns on antioxidant genes. So essentially it deals with redox. And BAC1 also activates AP1-like gene transcription. And these are the genes that actually lead it to promote metastasis. And you can see on the right, if you have cells with BAC1 and then you essentially deplete BAC1 using, in this case, SHBAC1, you can dramatically decrease metastasis, but then you can regain it by putting in these downstream genes essentially to rescue the original phenotype. But it's not enough to kind of do this in mice in a preclinical situation. We need to show that these players, ARCIT, the suppressor of metastasis, BAC1, the promoter, are actually clinically relevant. And so we looked at patients, and this is from TCGA. And as you go along the x-axis, you're looking at individual breast cancer patients, and you look at the y-axis, the top one is ARCIT, PEBP1, and then there's BAC1, and then these are a bunch of genes that promote invasion. And so as you can see on the top, if red color says this is expressed at a high level, so if you have ARCIT high, as we would suggest, BAC1 is low because it's inhibited by ARCIT, and so are the downstream genes that it might activate. If ARCIT is low, BAC1 is high, and so are the downstream genes. And then there are others that just don't play in the sphere. So we're not covering all of breast cancer patients. But if you look within TNBC, we can actually stratify TNBC patients and find the patients who have the worst outcomes. This is metastasis-free survival, and these will actually die faster if they have BAC1 and its signaling pathway expressed. So I want to tell you two stories in my remaining time. And the first one was done by a talented postdoc of mine who's now a professor at Georgetown, Jiang Li. And the question she asked is, okay, we know that BAC1 is a promoter of metastasis. So if we take it away, does that cause reprogramming of cells? We already know that that will suppress metastasis. But in the process, would it also increase sensitivity of these cells to drugs? And so we asked, if you take BAC1 away, what actually happens? What is it regulating beyond, let's say, invasion? And it turned out, to our surprise, that the major feature that it regulated was the cellular energy phenotype. That is glycolytic, aerobic, and oxidative stress, so metabolism. So if you remember your kind of basic metabolism, glucose is metabolized through a non-aerobic process called glycolysis to lactate, and then that undergoes oxidative phosphorylation, the process generating some reactive oxygen species. And this is the way it normally occurs, but in cancer, this is called aerobic glycolysis because glycolysis dominates. This was the Warburg effect that was identified by Otto Warburg hundreds of years ago. And since glycolysis is most important, but it also uses some oxidative phosphorylation, it's very hard to target this. If you target glycolysis, oxidative phosphorylation still generates energy. If you target oxidative phosphorylation, glycolysis is still very important. Why do cancer cells use this? Because glycolysis, in particular, even though it's less energy efficient, can lead to the intermediates that enable cells to make proteins, nucleic acids, DNA, RNA, and so on, that's needed for cell proliferation. So what happens when you remove BAC1? Well, interesting, it's almost like a seesaw. You move from a focus on glycolysis to actually almost entirely oxidative phosphorylation as a source of energy for cells, which is actually what happens in normal cells. And so looking at that, we thought, if one could identify a drug that could target oxidative phosphorylation, and as it turns out, metformin, which was a drug often used by diabetics, one of its effects, in fact, is to inhibit complex 1 in the electron transport cycle of oxidative phosphorylation. If we tried that, could it be more effective in a BAC1 low tumor versus a BAC1 high? Because now we've really essentially moved our heterogeneous tumor from the point of metabolism toward a more homogeneous tumor where metabolism is really occurring in oxidative phosphorylation space. And the answer is yes. If you partner metformin the inhibitor of oxidative phosphorylation, with loss of BOC1, you can have dramatic effects on tumor growth. So that's illustrated here, where if we look at tumor growth, here's the control. If we put in metformin, really it's unchanged. If we knock out BOC1, it's unchanged as well. But if we combine a knockout of BOC1 or knockdown of BOC1 in this case with metformin, then actually we get a dramatic suppression of tumor growth. So what does this suggest? It suggests that we can target molecules like BOC1, which are regulators of metastasis, in fact, promoters of metastasis, as potentially a new therapeutic strategy. But what have we actually done? As I just alluded, if you look at the phenotype of metabolism, energy generation, generating essentially the units for biosynthesis, promotion of cell growth, then if you start out with a heterogeneous tumor and you target it with, let's say, targeting BOC1, and in this case, we're actually fortunate because there's actually a drug that can target BOC1 as a transcription factor. It's very unusual. Usually they're really untargetable, and that's heme. We could turn this into a more homogeneous tumor from the point of view of metabolism, then treat it with the drug metformin. And now, because the cells are more uniform and they're more in oxidative phosphorylation space, we can effectively suppress tumor growth. So this seems pretty good. And it's a strategy we're still pursuing and actually demonstrating on the level of single cells to show this really happened. But there's still a major challenge in this metastatic space. And that is that single drug treatment is largely ineffective. So although targeted drugs that work in theory have been identified, this heterogeneous and evolving state of the tumors often makes these approaches ineffective. So the challenge we face is to identify synergistic drug combinations or treatment protocols that eliminate the ability of these tumors to adapt and suppress metastasis. And we can illustrate it this way. So if you look at a cell network, it's pretty complicated, but you can imagine a cell network is a little bit like a roadmap where one's moving from one city to another. So imagine we want to go from Memphis to Miami. If we were talking about a disease like heart disease or even HIV, then the pathway is pretty linear. It's pretty much one road. And so we can put a blockade up along that road, and we can block 90, maybe even 99% of the people getting through, but somebody is on a motorcycle, manages to get around. But then we put up another blockade. And eventually, if you put up 2 or 3 blockades, you can prevent everybody from getting through. But in cancer, if you're going from Memphis to Miami, you can go anywhere. You can do a workaround. You can go to Boston before you go to Miami. So blocking one road is not sufficient because there are other effective roads. We can call them signaling pathways, or as I'm going to get to, even cell networks that actually enable you to do a workaround. And that's why cancer often recurs, even though initial drugs are effective. So what do we need to do? We need really to put, let's say, a moat around Miami so no cars can get across. It's blocked. How can we do that? So addressing this question, a really talented former graduate student in my lab, Ali Yezelkanal, asked this question. Well, what if we reprogram metastatic cells and try to turn them into non-metastatic cells by mimicking RKIP, the metastasis suppressor that we've already identified? So we already know RKIP does it. It's a natural regulator of metastasis. So can we come up with a drug combination that does what RKIP does? Because unfortunately, we don't have a good way of introducing RKIP itself into breast cancer cells in people. So to address this question, Ali first turned to an approach that a colleague of ours, Gary Johnson at the University of North Carolina, developed. And it's called this MIB-MS approach. And basically, what it's about is he utilized inhibitors of active kinases. And I told you in this case, kinases are actually the targets of RKIP. And they trap the kinases when they're in their active state on beads. So he basically fixed the inhibitors to beads, trapped them, and then pulled out these active kinases, put them in the mass spec, and quantified them. What kinases are actually active in state A or state B? And state A is the control, and state B is a state where we've now introduced RKIP, and we've suppressed key kinases that control effectively our ability of cells to undergo metastasis. So what are they? So as you can see from this heat map, where red is high and blue is low, after putting an RKIP, most of the kinases, their activity goes down. But in a few, the activity goes up. So we focused on the ones where the activity went down. And then we built a network out of it, a kinase network. And what we discovered that, and again, this will be a little bit cell type specific, so we used triple negative breast cancer cells that actually had mutations in RAS and in RAF. And so it's not surprising that this pathway leads to a very common pathway called the MAP kinase pathway. And it turned out the core of this network are three key MAP kinases called ERK, which was the original one that was identified, JNK, and in this case, JNK1 and 2, and then P38. And interestingly, JNK and P38 were identified as stress kinases. And I told you that stress is linked to metastasis, so that makes sense. And ERK, even though it's known to regulate growth to a large extent, can also contribute to a stress response. So we asked a couple of questions about the way RKIP acts, trying to get some kind of intuition. What is it doing? So initially what we thought was, okay, these are kinases. This is a really aggressive tumor. So if we target the most active kinases, that must be what RKIP's doing. So let's stratify these kinases according to activity, where red is high and blue is low. Won't we find all of these metastatic kinases kind of at the top in the red zone? And MAPK1, which is ERK, is right there, just as we might have expected. But interestingly, most of the kinases don't sit there. So you can have different levels of kinase activity, but as long as they're contributing to the network, they are key and important players. But more importantly, when we asked, what is the level of inhibition that RKIP is able to manage for each of these kinases that we're observing? So as we're going along, we're looking at the percent of activity that's changed, that's reduced in the case of the kinases whose activity is suppressed. We discovered that it's never more than about 30%. You never get up to 100%, which is what we're always trying to do when we target tumors. We play what's called a whack-a-mole game. We see a kinase, like let's say RAF kinase with a V600 mutation, and we use a drug and we hit it and we wipe out RAF kinase, and that disease is particularly known for melanoma, is gone for about 7 months, and then it comes back with a vengeance. Another mole came up in a different place. There's another mutation. And so we have to hit that one. And so often our treatments are following the mutations. We get one, another one pops up, we get another one, and so on. And as you can see, this can basically end up as a no-win game. That's not what RKIP does. So we thought, oh, what if we had a combination of inhibitors that focus on the similar kinases that RKIP's targeting and see whether we can mimic it with this combination? So what we first did was work out the network for one cell line TNBC that we were using. These arrows mean that this kinase is activating this kinase. So MLK could be activating P38, RAF, very well known, activates MEK, which activates ERK. And then you have other interactions such as GNK to ERK here, or tau-Ks to ERK, and those are inhibitory. And those are considered kind of feedback regulation. So we have positive regulation and negative regulation. And then we said, all right, what happens if we start to target all of these key kinases? And we can do them as single. We can do them together, double. We can do them in triplicate, and so on. How well do they work? And we screen them in two ways. We looked at growth, and our goal here, remember, we're trying to target metastasis. So we weren't necessarily interested in suppressing growth. So we deliberately used low doses to make sure we weren't getting toxicity or effects on growth. But at the same time, if you're looking at cells in culture, then the phenotype you're looking for that seems to be a correlate to what we see as metastasis in vivo is invasion, the ability of cells to not only move, but actually chew up the matrix around them and migrate through it as though they were connective tissue in the body. And so our goal was we want to get invasion down, but we don't want to significantly affect growth. And interestingly, this was not just a linear thing. It wasn't that 2 drugs were better than 1 and 3 were better than 2 and so on. Sometimes 2 drugs were better, 3 were worse, 4 were better. In the end, we ended up with a 4-drug combination. We inhibited MEK, JNK, P38, and MLK. So down here, this guy, JNK, P38, and MLK. And then when we tested it, we used 3 different TMDC cell lines, 2 BM1 and 436. We're from human and LMB is from mouse. And the goal was we needed to suppress all the key MAP kinases there. And so if you look at each of these, the red color is you start out in the control and they're all active. And where this arrow is, if you do that 4-drug combination, you see that they're all inhibited, which is not the case when you use other combinations. So we thought, oh, okay, let's try that in mice. And so this is one example where we tried it in mice. And in this case, we were using luciferase labeled cells, so they light up with photons. And we injected them such that they would metastasize in the way I showed you earlier. And we monitored the metastasis. And what you could see is the control, very metastatic. If we used a kind of half dose of this 4-drug combination, it was actually pretty effective, and a full dose was even more effective, and this is plotted here. You can see you got almost complete suppression of metastasis. But what was really encouraging was that if we did a 3-week treatment and then we stopped and we kept going, you could actually get survival for at least another, in this case, about 30 days. So you could double the survival of the mice by doing this treatment. So the downside is you probably have to keep doing a treatment like this periodically, but the upside is it was quite effective. So the question we wanted to address is why? Why did this work? Why does RKIP work, actually? What's going on? And there are 2 key reasons. And so that means I need to break it down in 2 ways. So the first question is, why did we have to do more than 1 drug? Why a combination? And in part, the answer is, well, you actually don't necessarily if you're talking about a single cell. The reason relates to the fact that we're talking about different cells that are not homogeneous. They have different networks, and we want to cover them all. So what's the example? So I started out showing you this network on the left for this 1 cell type BM1, and I'm going to focus on a small aspect of it, of the 3 MAP kinases. If you look at their interaction, P38 activates ERK and JNK inhibits it. If you look at a second cell line, 436, also human, well, it's opposite. P38 inhibits ERK, JNK activates it. And now if we look at this mouse LMB, P38 also inhibits ERK, but JNK really doesn't do anything. So now what happens when we look at the ability of these kinases to act on these cells? And in this case, we're using BOC1, which I told you is promotive metastasis, as our kind of stand-in for invasion and metastasis, since these kinases activate it. And so using BOC1 as a readout in the BM1 cells, turns out P38 does lead to BOC1 activation, and ERK does. So if you used inhibitors for those, they could be effective, but JNK doesn't. And yet, if you use the 4-drug, it works. In 436, P38 and JMK can lead to BOC1 activation, but ERK doesn't. So if you use that inhibitor, it wouldn't work, even though the other two might, but the 4 drug works. And LMB, JNK and ERK can activate BOC1, but P38 doesn't do anything. And yet the 4 drug works, although I'll say here that it's not quite as potent. So I think this is the most interesting aspect of this study. The previous point is that you want to have multiple targets, just because you're looking at multiple different types of heterogeneous cells that you're covering. And so you're actually, in effect, covering a network and not just a single target or a single pathway. But why did we end up with a low dose? Why is that important? Which is not the way we're generally treating patients now. And the answer comes again from going back and looking at the network. So if you look at a receptor, such as the epidermal growth factor, EGF receptor, it activates this little cascade, which starts with RAS. It's inactive until it's bound to GTP, and then it's active. And when the phosphate is removed, it goes to an inactive state. But when it's active, as I alluded to before, it activates a very well-known and, as it turns out, conserved to yeast pathway of RAS, which is a G protein, to RAF, which is a kinase, which activates MEK, another kinase, and ERK. So this is a MAP kinase, this is a MAP kinase, and ERK is a MAP kinase. Well, that's not the only thing that RAS does. It also activates other pathways, like PI3 kinase going to AKT, another kinase, and then mTOR, which leads also, both of them, these pathways, to proliferation and potentially invasion. So why would we have them both? And the answer is we don't. If one of them is highly active, like the RAS-RAF-ERK pathway, then actually there's a feedback from ERK to AKT, and it shuts off the other pathway. But now, if we use the MEK inhibitor and inhibit that MAP kinase pathway, this feedback inhibitory interaction goes away, and now you activate a different pathway, effectively a different network. So how does that play out experimentally? So if we drop EGF on a cell and we're now monitoring ERK activity using phosphorylation as a measure of it, we can see that as we're adding the MEK inhibitor, ERK goes down as we expect, but AKT goes up. And it goes up in an interesting way. It's not linear. It's actually a threshold effect. Where does it begin to go up? Just at the point where you're inhibiting about 20 to 30%. After that, if you go beyond that, it shoots up. So what's the space you'd like to be in? You'd like to be down here, where you're starting to inhibit ERK, but you're not yet activating AKT. And so when we looked at tumors with the 4 drug, we saw that yes, we were inhibiting ERK with the 4 drug, but we were not activating AKT. So it's exactly what you want and where you'd want to be. So how can we think about this a different way? So think of it as a faucet, a spigot, and it's pushing out water. And you put your hand up, and if you're just suppressing the whole thing, and this is just like signals coming through a signaling pathway, it's got to go somewhere, and it can go in the same direction and activate another pathway. In this case, let's say the AKT, PI3 kinase AKT, that's going to give you resistance and recurrence. But when we actually decided to only inhibit about 20 to 30%, and then we don't have only one spigot, we have 4, so we can do that 4 times, the water doesn't all go in one direction. It just dribbles through. So some of it comes through and it gets dissipated so that essentially it goes away. And now you don't have a compensatory network because it's not all going in one direction. You don't get resistance and you don't get recurrence. So to summarize, we've kind of built up a network. We've shown that not only does a metastasis suppressor do this, but so does a 4-drug MAPK inhibitor of MAP kinase that leads to BOC1 invasion metastasis. and that it's effective, seems to be well tolerated and robust anti-invasive therapy. And we think that targeting multiple nodes associated with distinct pathways, but within the same network, is likely to improve. So how does this relate to what we do? So we do sometimes, as I said, in combos, vertical treatment, which is take a single pathway. But if we knock these down, the feedbacks now don't work, and then you get activation of different pathways. And so in the end, you get high toxicity and compensatory activation. Same thing horizontally. If you go across and get multiple pathways, but you inhibit one or inhibit another, then the ones that they're suppressing can be activated, and it could also give you high toxicity and compensatory activation. But we would suggest that tamp it down. Don't get rid of it. Don't push the cells and the signals to have to move to another network. Just suppress it a bit, dissipate that signal, and it will be effective. So what are the next steps? We want to selectively target genes that respond to stress and promote heterogeneity, such as SPOC1. So we want to do that a little better and more selectively. We want to refine drug combinations that target metastatic networks. 4 is a lot for clinicians, so we want to lower that down. We want to develop gene signatures and biomarkers that enable selective patient identification to increase treatment efficacy. And finally, the immune system is the most dynamic system that we have in the body. And so we want to partner our drugs with the immune system and essentially reprogram tumors to increase the ability of immune cells to be effective, to infiltrate tumors and to work. And so with that, I'd like to thank the people in my lab, particularly previous colleagues, Jiang Li and Ali, who I mentioned, and Marguerite, who's helped me with all the clinical parts, our previous collaborators, and this is my lab. And basically all the work comes from my lab. It's not from me. And they're the ones who really get the credit. And finally, the institutions that support us. So with that, I'd like to thank you and answer any questions that you might have.
Marsha, thank you so much. That was such a fantastic presentation. And before we jump into the Q&A, because we do have quite a few questions rolling in, I just want to remind the audience, if you do have questions for Marsha, please submit them in that Q&A tab on the right of your screen, and we'll try to get to as many as we can today. And also, if you do have to leave early, I am going to launch a little survey pop out here. If you could give your feedback in that, it'd be greatly appreciated. All right, and with that, now I'm going to bring Marsha back on here. There you are.
I can't see myself, but that's okay.
That's okay. We can see you. Again, thank you for that fantastic presentation. Really, really cool work, and excited to jump into these questions. So the first question here is, if low dose targeting is effective in the mice studies and dosing is seemingly very important, how do you determine / titrate this precise dose for translation to clinical studies?
So that is a great question, and that's something that we're actually trying to address now. So in mice, the way we did it was to try to determine the dose that's beginning to suppress the growth. In fact, that actually didn't work. And to our chagrin, we not only blocked metastasis, we blocked growth, which is what everyone else is trying to do. We were actually trying to be selective to say, oh, let's combine this with something else that's cytotoxic and can block growth. So we actually know the highest tolerated doses for each of these drugs. And the idea is to take them down in a way that's parallel with what we can do in the preclinical studies. So some of this will be some trial and error, but our hope is that we can do this on models like organoids that actually come from patient tumors or patient-derived xenografts, so that we can really test it in models essentially before we get to people. But I'll also say that it's possible to dose people and look at their tumors early on to see whether you're successful in suppressing these pathways as you would predict. So we can also monitor patients as we're treating them.
That's fantastic. And it leads right into the next question that I have here for you. This question is, there seems to be an increasing trend towards personalized medicine in oncology. Are there possible applications of this sort of approach here with regulators of metastasis in breast cancer?
Absolutely. And that's what I alluded to at the end. So what we're now doing is there's a real revolution in our ability to look at cancer, which is we can now look at cancer on single cell level, even in patients. And so what we've been doing is developing gene signatures. And at the moment, these are based on mRNAs, but downstream of the regulators. So you can actually use these signatures to know whether the regulator you're interested in is active or not. And then we can use these now to begin to identify patients, and particularly their tumors, that might be susceptible to an appropriate treatment that would target that regulator. So that's what the intent is for everyone really doing now targeted therapy, is to match the patient with the therapy and not just do this kind of more generally.
Absolutely. Thank you so much. We have another question here. I know breast cancer in males is much more rare, but are there gender differences in these mechanisms and treatment efficacy?
Yeah, I think that's a great question. It's totally the case that males can get breast cancer. And in fact, in all of our research, we're asked to justify the kind of mice that we use. Usually it goes the other way around. Everybody uses male mice and they're saying, hey, can you throw some females in here to see whether they have the same effect? I'll give you a very simple answer to that, which is I personally don't know because we have focused at the moment on female. But I would imagine there are a lot of similarities in terms of the basic mechanisms, but some of the details will probably be different.
Thank you. Yeah, definitely more rare, but still important to address.
Absolutely.
All right. We've got another question here. Do these sorts of metastatic approaches work in other cancers as effectively, or are they more effective in breast cancer specifically?
Okay, again, a great question. So when we did that original study on metabolism, we actually did an analysis of data from other patient tumors. And what we found was that in a fair number of tumors, pretty common tumors like prostate and pancreatic and so on, the same relationships existed. So BOC1, again, was pushing cells into a glycolytic state. And so the answer, we believe, is that targeting it in some of these other tumors would also work. But then we also have found a couple exceptions. So we think most tumors follow that. But one of the exceptions, interestingly, was lung tumors. And we know that BOC1 plays a key role in metastasis in lung. Interestingly, even in combination with NRF2. NRF2 kind of does the job of making the tumors grow and survive. And then BOC1 makes them metastatic. But interestingly, NRF2 has another regulator called KEAP1, and that plays a bigger role in lung. And so you don't find these quite as linear inverse relationships, let's say, or even direct relationships, correlations between BOC1 and some of the outcomes because there are more players there. So there is a cancer specificity. But in general, we think that this has wide application. But more than that, even though I talked about two regulators, what I'm most interested in are the principles. What are they actually doing? And we know that there are other players that can do that as well. So it's the principles that can be applied, even if not always the particular targets.
Amazing and so complicated. Thank you so much. I just realized the time, and that will be our last question for today. I just really wanted to thank you for your expertise, for delivering such a fantastic presentation, and for answering all of these questions. It really was a pleasure to have you with us, and I hope that you had fun.
Thank you. And I really appreciate the opportunity to do this, to give my talk and share my work. So thank you all for listening.
All right. And I did want to thank the live audience as well. We know you have busy schedules. We really appreciate you being here live and hope that you enjoyed your experience today. Please join us for the next event in the series in 2 weeks on Wednesday, February 19th. This event will be at noon Eastern time. So all of the previous events have been at 11. So just make a note of that. It'll be at noon Eastern time, and you'll receive reminder emails just like you have for the rest of the series. So keep an eye out for that email. Another quick reminder that a recording of today's event will be available soon, so also keep an eye out for an email giving you access to that if you want to watch the recording. And additionally, a survey will appear at the end of this webinar. We do read all of your comments and appreciate your feedback, so we would love to hear from you. And finally, if you still have questions for our speaker, you can submit them in the survey and we'll be sure to pass them along. In closing, we hope that you enjoyed this Inside Scientific webinar produced in partnership with the American Physiological Society, and we'll see you in 2 weeks.